Which enzyme is responsible for relieving supercoiling tension in DNA during replication? A. DNA polymerase. B. Topoisomerase. C. Helicase. D. Ligase. The correct answer is B. Topoisomerase is responsible for cutting and rejoining DNA strands to relieve the tension caused by supercoiling as the DNA unwinds during replication. What is the role of the sigma factor in bacterial transcription? A. Termination of transcription. B. Recognition of the promoter. C. Elongation of the RNA strand. D. Splicing of introns. The correct answer is B. The sigma factor in bacterial transcription helps the RNA polymerase recognize the promoter region of a gene, allowing the initiation of transcription. Which of the following components is not involved in the formation of the eukaryotic transcription initiation complex? A. Tata box. B. RNA polymerase II. C. Rho factor. D. General transcription factors. The correct answer is C. The Rho factor is involved in transcription termination in prokaryotes, while eukaryotic transcription initiation involves the Tata box, RNA polymerase II, and general transcription factors. What feature distinguishes euchromatin from heterochromatin? A. Presence of repetitive DNA sequences. B. Greater transcriptional activity. C. Denser staining during interphase. D. Lack of histone proteins. The correct answer is B. Euchromatin is transcriptionally active and less condensed than heterochromatin, allowing genes within it to be more easily accessed for transcription. The enzyme telomerase is essential for A. Extending the 5 ends of chromosomes. B. Repairing mismatched DNA bases. C. Adding repetitive sequences to the ends of linear chromosomes. D. Splicing out introns from pre-mRNA. The correct answer is C. Telomerase adds repetitive nucleotide sequences, telomeres, to the ends of eukaryotic chromosomes, protecting them from degradation during DNA replication. Which of the following codons is considered a stop codon in the genetic code? A. AUG. B. UGA. C. UAC. D. AAA. The correct answer is B. UGA is one of the 3 stop codons, along with UAA and UAG, that signal the termination of translation by the ribosome. Which of the following is responsible for the splicing of introns from pre-mRNA in eukaryotic cells? A. Ribosome. B. Spliceosome. C. RNA polymerase. D. DNA polymerase. The correct answer is B. The spliceosome is a complex of proteins and small nuclear RNAs that catalyzes the removal of introns from pre-mRNA, producing mature mRNA. The Meselson-Stahl experiment demonstrated the semi-conservative nature of DNA replication by using A. 14C and 12C isotopes. B. 15N and 14N isotopes. C. 32P labeling. D. Radioactive sulfur and phosphorus. The correct answer is B. Meselson and Stahl used 15N and 14N isotopes in their experiment to show that each newly synthesized DNA molecule consists of one old strand and one new strand. During translation, the peptide bond between 2 amino acids is formed by the catalytic activity of A. tRNA. B. rRNA in the ribosome. C. RNA polymerase. D. DNA helicase. The correct answer is B. The ribosome's rRNA, specifically the 23 srRNA in prokaryotes and 28 srRNA in eukaryotes, catalyzes the formation of peptide bonds during protein synthesis. Which component of the ribosome binds to the mRNA first during the initiation of translation? A. Large ribosomal subunit. B. Small ribosomal subunit. C. Aminoacyl tRNA. D. Peptidal transferase. The correct answer is B. The small ribosomal subunit first binds to the mRNA at the start codon, AUG, during the initiation of translation, after which the large subunit associates. Which process ensures that the genetic code is translated accurately during protein synthesis? A. DNA replication. B. tRNA charging. C. RNA splicing. D. Topoisomerase activity. The correct answer is B. tRNA charging involves the attachment of the correct amino acid to its corresponding tRNA by aminoacyl tRNA synthetases, ensuring accurate translation of the genetic code. Which of the following regions of DNA is most likely to be transcribed into mRNA? A. Enhancer region. B. Promoter region. C. Exon region. D. Intron region. The correct answer is C. Exons are the coding regions of DNA that are transcribed into mRNA and later translated into proteins, whereas introns are spliced out during RNA processing. Which of the following mutations would likely have the most drastic effect on the resulting protein? A. Silent mutation. B. Missense mutation. C. Nonsense mutation. D. Frameshift mutation. The correct answer is D. A frameshift mutation alters the reading frame of the mRNA, potentially changing every amino acid after the point of mutation and leading to a non-functional protein. The term wobble in the genetic code refers to the ability of A. Ribosomes to skip over stop codons. B. tRNAs to recognize multiple codons for the same amino acid. C. RNA polymerase to backtrack during transcription. D. DNA polymerase to correct errors in replication. The correct answer is B. Wobble allows certain tRNAs to recognize more than one codon due to flexible pairing at the third nucleotide position, ensuring efficient translation despite codon redundancy. Which type of mutation results in a premature stop codon? A. Missense mutation. B. Nonsense mutation. C. Silent mutation. D. Frameshift mutation. The correct answer is B. A nonsense mutation changes a codon for an amino acid into a stop codon, resulting in the premature termination of translation and a truncated, usually non-functional, protein. Which of the following enzymes is involved in the removal of RNA primers during DNA replication in prokaryotes? A. DNA ligase. B. DNA polymerase I. C. DNA polymerase III. D. Primase. The correct answer is B. DNA polymerase I removes RNA primers in prokaryotic DNA replication and fills in the resulting gaps with DNA nucleotides. Which histone protein is not part of the nucleosome core but helps in the compaction of chromatin? A. H2A. B. H3. C. H8. D. H4. The correct answer is C. Histone H1 is not part of the nucleosome core but helps in stabilizing the chromatin structure by binding to the DNA between nucleosomes, aiding in further compaction. Which of the following is the main driving force for DNA replication to occur in the 5 to 3 direction? A. Energy released from ATP hydrolysis. B. Formation of hydrogen bonds between bases. C. The activity of DNA helicase. D. The 3O group required for nucleotide addition. The correct answer is D. DNA polymerases can only add nucleotides to the 3O group of the growing DNA strand, ensuring that DNA replication proceeds in the 5 to 3 direction.